recording. I'm going to share the screen. I warned you before, I'm not sharing my screen, and I see somebody's done a chat. I don't see the messages, so if you... Oh, that's fine. You're just asking other students to study together. All right, let's share sound and share the screen. Now, I logged in as a student, so I might be able to see how to do this. So, Worksheet Chapter 1, and you click here, and it gives you the worksheet. And let's say you want number 1. So, naming a classifying organism was begun in the 18th century by, and you scroll to the bottom and you see where it says reply. Hit reply, and then type your answer. I spell his name, Linnaeus. And then
so
click
to only be for one
click
question?
post reply.
Is it supposed to only be for one question then?
Yeah, you want to answer another question. Somebody's already answered question 1, so if you're the next person in, you answer question 2. And why is it only one question? Because we are a large class, and even if people only answer one question, there are only 33 questions, and there's more than 33 people in this. It's 2 sessions. Okay? And so, obviously, when the 33 questions are answered, what you're going to have to do, and I'll mention it now, is you'll need to start at question 1 again. You could also look for one you think is wrong and answer that question. Okay?
Okay, I understand now. Thank you.
All right. Yeah, I was able to show you when I went into student view. Now, how do I end student view? I might have to. I don't remember. Go home. Now I'm locked into student view. There's a way to end it, but I'm sure not finding it. Oh, here it is.
It says
down
on
here.
the bottom.
Yeah, right there. Leave student view, sorry. There we go. All right, now let me bring this down. I might mute myself by touching that. Can you hear me?
Yes.
Okay, good. All right, let's go on to just briefly the syllabus. I've already talked about the syllabus in the lecture. We have the same syllabus in the lab. I need to check on this. I don't think that's right, but I'll check on that. If you contact me, the best way to do it, I didn't mention that, is to contact me with the Canvas email, because I'll only get student emails in the Canvas email. In the other email, I'll get other emails. And I do have a Clark College email, which I should put on my syllabus. I didn't realize I don't have that there. It's something like jvolpe @instructor@yavapai, something like that. I'm just going to really talk about the schedule here. Oh, no, there's one other thing I need to talk about. Laboratory safety. I've already talked about the grading. It'll be together with the lecture, so I'm not going to go over that again. But there are some projects which are put into the lab. 2 major projects, the infectious disease research project and the unknown project. All right, let's talk a little bit about safety, lab safety and rules. You do not need to know them in a sense because we're not actually going to be in the lab. Everything will be online. But safety is one of the main skills that students learn from a microbiology lab. So you do need to know this material and you will be tested on it. Don't take shortcuts because those risk the safety and accuracy of what you do normally in the lab. And when you were to take a shortcut, you could contaminate the environment that you're working in, as well as you and other students in the lab. So nothing is taken into the lab because that can bring in things, and anything that is brought into the lab, like a pencil or a pen, has to stay in the lab. And you cannot take them out of the lab until they are treated, either in bleach or in the autoclave. And if you were to take out your phone, for example, in a real microbiology lab, it would be confiscated and it would be put into bleach or autoclave before being returned to you. And guess what? That would destroy the phone. So don't do that. Only enrolled students are allowed in the lab. Personal items can be brought in a book bag and then be left at the cubbyhole by the door. So the region where the door is and the cubbyhole, that's where you put things in a lab. That is considered outside the lab. And then you walk into the lab, which you're already actually in the lab. But the point is that region is considered outside the lab, so you can bring things in and leave it in the cubbyhole. You should put all your books and everything there, like your coat. You don't take that to the lab, because if you take that to where you sit, that would be confiscated and you would not get it back until it had been treated by bleach or in the autoclave. Somebody had a question.
Yeah, are we supposed to be like going to the college or something for lab, or do we have to purchase something? I'm not really understanding how we do all this from home.
Our lab is going to be done online. I'm telling you, you need to know about lab safety in a real lab, and you will be tested on it, but you're not actually going to go into a lab. Okay?
Okay, thank you.
All right. All cultures can be potentially pathogenic, so keep hands, pencils, and other materials away from your mouth, your ears, your nose, etc. No eating or drinking or smoking or gum chewing or any other oral habit is allowed in the lab. You have to wear gloves while you're in a microbiology lab, at least in my lab. You should monitor yourself if you get infected and then have a like diarrhea that could be caused from the lab. There should be wearing a lab coat while you're in the lab. And when you come into the lab, you should wash your hands after you put away your books. Wash your hands and put on gloves, put on your lab coat. Okay? And then go to your workbench, and actually you should then clean or disinfect your work area, your workbench area. And then when you're ready to leave, you should clean the workbench where you worked to kill all the microorganisms, and then take off your gloves, take off your lab coat, go wash your hands, and then you can leave the lab. Any question about that? So before you work in the lab, you should clean the lab bench, and then after you're done working and before you leave, you should also clean the lab bench. You should note where fire extinguishers are. There's a fire blanket, an eye wash station, a first aid material. You should know where those are in the lab. Obviously, we won't ask you about that because you won't be in the lab. You should report accidents like spills to the instructor and then clean them up. If there's broken glass, take care. Let's talk about a broken glass spill. Cover the material with paper towels, then put disinfectant down, let it stand for 15 minutes, wipe it up if needed. If there's broken glass, put the broken glass in the broken glass container, put all of the other material in the biohazard bag, which I also call the autoclave bag. The biohazard bag has contaminated waste that is not glass that will be autoclaved before it is disposed of. Broken glass we put in the broken glass container. We never put glass in the biohazard bag. Like if you're working with a glass petri dish, it should be put in a place in the back of the lab that we store the, not the glass petri dish, the glass test tube, sorry, the glass test tube. The petri dishes and anything that's plastic can be put in the biohazard trash. Anything that doesn't have microorganisms on it, like a paper towel that, I don't know, you're wiping, I don't want to say oil off the microscope, like on the side of the microscope, or water got on the side of the microscope, you use a paper towel, there's no microorganisms on it, that goes in the trash can. But if there's microorganisms on it, put it in the biohazard thing. For working in the microbiology lab, we have tools, and we would use a Bacto Incinerator. I'll try and show you that later. It'll be in the first lab. You put the tool in the Bacto Incinerator, and it will sterilize the loop. And I'll show you what the loop is when we get into lab 1. You should have your long hair tied back or put in a rubber band so it doesn't fall into the Bacto Incinerator and then catch fire. No mouth pipetting is allowed in the lab. Never take cultures out of the lab. If samples are brought into the lab, you have to get authorization from the instructor. So nothing really is brought into the lab, and certainly nothing's ever taken out. If you have a cut, an open sore, you should wash your hands and then put on a Band-Aid. We have Band-Aids in the lab, and then put the glove on top of the Band-Aid. All cultures should be labeled completely with your name, the date, the name of the specimen, the medium, and optionally the temperature can be added. We'll talk about that more in the lab specifically. So agar petri plates are going to be plastic. They're put in the biohazard bag. Glass slides and cotton swabs are put in the glass container with disinfectant. There will be one for glass slides and one for cotton swabs. You can find them in the rear of the lab. Sometimes they're also on the lab tables. Pipettes, if it's glass, we put it in the container labeled pipettes. Generally, my students don't work with pipettes, and if they do, they work with a plastic pipette. The plastic pipette can go in the autoclave bag. Test tubes, remove the labeling tape, like if you put a name on it and a culture on the tape, remove the tape and then put it in the test tube rack in the back of the lab. Supplies are found in the front of the lab. We can skip that. I've already talked about them. And you have to comply with the safety procedures in the lab and COVID-19 while you're at Yavapai College. And you are required to follow these rules. Normally I could kick you out of the lab if you do not, but like I said, you'll be tested on this material. Any questions about any of that? All right, for today, oops, I just went past it. We're going to be doing Lab 00, the laboratory safety and training. This will be the first lab. You also need to read about the infectious disease project. That's the file on Canvas under the first lab. Let me show it to you. I'm going to talk about it on Thursday, so you need to read about it. So we scroll down to the lab. There's the worksheet for Lab 00. There's the Lab 00. And there you need to read about the infectious disease project. These will be done on Wednesday. Okay? And I've got a reminder here. You need to take the plagiarism quiz by the end of this week. And that is up here. Plagie quiz. You have to take the quiz by June 11th. There is a tutorial which you should take, or at least get started before June 11th. It isn't short, the tutorial. I don't remember how long it takes, but it might be, it's at least a half an hour. It might be an hour long, the tutorial. And then there's a fun video about plagiarism. It's optional. It's made by Yavapai, whereas the plagiarism tutorial is not. All right, any question about any of that? If there's no questions, let me remind you, on Wednesday I'll discuss the infectious disease project. So you should read about it. The infectious disease project is not due until Friday, June 11th, but we'll discuss it on Wednesday. All right, let's talk about the lab. Lab Module 00, Laboratory Safety. You're not going to be actually working in the lab, but you do need to know this material as the rules and procedures. You will be tested on them. Upon successfully completing this laboratory, students should be able to distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab, and understand how to safely dispose of contaminated materials. Any question about that? All right, you are expected to follow certain safety rules while you're in the microbiology lab, and that is to handle potentially pathogenic microorganisms, and every organism you handle should be considered potentially pathogenic. Even E. coli, if you were to get enough of it, would make you sick. And obviously that's if you were to consume it. If you got the E. coli someplace else than your gut, and like I said, it would make you sick even if you got it in your gut. But if you got it in other locations, it could grow in those locations and make you sick. So everything is considered potentially pathogenic. You're not actually going to perform any of these activities. You're all going to be doing it online. For safety, when you're in a real microbiology lab, you should wear proper attire that includes a clean lab coat, and the lab coats are kept in the lab, and every term they are washed and autoclaved. And I think they're autoclaved first and then they're washed, but it could be the other way around. I don't remember. Once you have your lab coat on, you should put on disposable gloves. When you come into the lab, you should only have closed-toed shoes. Sandals, thongs, a woman's shoe that's not closed-toed is not allowed, because if you have a spill, the microorganisms will get on your feet and then you will take them home. So we require closed-toed shoes to give you some protection from the microorganisms you're working with. When you're working with a splash hazard, you have to wear goggles while in the microbiology lab. Fortunately, we don't often work with splash hazards, but if you're working like staining a slide, or you are working with a liquid culture where there's microorganisms in it, that you could splash into your eyes, and you have to wear goggles. And then you remove all of these things before you leave the lab, and they remain in the lab. The goggles will be put into a container to be sterilized with UV light. The lab coat will be hung up in the lab and won't be used again until you come into the lab and use your lab coat. The gloves, of course, are thrown away and you use new gloves. Additionally, the hands must be washed shortly after you enter the lab and shortly before you leave the lab. So that'll be the first thing you do once you're in the lab, and then the last thing you do when you leave the lab. Long hair should be tied back. This prevents it from catching on fire and falling in the microbes. No food or drinking in the lab. No use of electronic equipment. No personal items are allowed in the lab. If you bring them into the lab, they will be confiscated. Like you could bring in a pencil or a paper, but then that pencil or paper or whatever you bring in the lab must remain in the lab until it is treated with bleach or autoclave. And obviously paper and pencil can be autoclaved and the pencil can be bleached. Alright, so a picture of what you should actually be looking like, and this looks something like our lab. You should have closed-toed shoe, wear a lab coat which is fully buttoned, wear lab gloves, and I don't know if you can see it, but there's goggles there. We do have goggles which can fit over eyeglasses, so if you have eyeglasses, we have big goggles that will fit over the eyeglasses. Keep your work area clean and uncluttered. Before you begin, you should sterilize or disinfect the area you work. That's to prevent microorganisms from getting in your cultures and contaminating your cultures, as well as when you leave, you should disinfect to kill any microorganisms that you may have contaminated the surface with. It may not be a spill. It could just be that you opened the dish and some microorganism floated out of the dish and went onto the table. Anyways, you clean the area before you leave so there's no bacteria on the area. And that's actually a picture of our lab, if you were to come to the lab, which we're not going to do that. Discard waste in the appropriate receptacles. Regular trash that's non-contaminated, no sharp objects. Gloves that don't have bacteria on them can go in the regular trash can, which isn't shown here. It's a normal trash can which we have in the lab, like paper. No glass ever in a trash can or in the biohazard container. Anything that is contaminated with microorganisms should be put in the biohazard container. Also, I call it the autoclave container. Okay? Like if you spill microorganisms on your glove, when you take the gloves off, put them in the biohazard container. No glass goes in the biohazard container. So if you have broken glass, it goes in the broken glass container, which I don't think that's shown. No glass containers, which we don't really use beakers like this, but we do use test tubes. Those go in the back of the lab in a container to put used test tubes in. And you should remove the tape from the test tubes. Use swabs. Go in a container marked swabs, and it'll have disinfectant in it. We have a container both for slides and cover slips. Remove the cover slip from the slide, put it in the container for the cover slip. We throw the cover slips away because they're so fragile, we don't wash them. The slides, put it in the container marked slide. It'll have disinfectant in it, and those will be washed and reused. I think I've already stated that. Everything that's contaminated, go into the autoclave or the biohazard bag. If you're unsure how to dispose of something, ask your instructor. Report all spills and accidents immediately, no matter how slight. Report it to the instructor. Obviously, you won't be in the lab, so you won't be doing that, but if you were to be in the lab, you should do that. You need to know that. Cover the area with a paper towel, like here, and then put disinfectant on it to kill the microorganisms. Let the disinfectant soak for 15 minutes before you pick it up, and then put this in the biohazard container. For other students, like if this were on the floor, walk around this area. You should keep lids on all petri dishes and bottles when not in use. That's to prevent them from being contaminated. Never mouth pipette. Never put anything in your mouth, including your fingers. When you're done with the Bacto Incinerator, we don't use a Bunsen burner, we use the Bacto Incinerator. Turn it off. When you're using it, you can have it on, and when you're going to be using it, you can have it on. We use the Bacto Incinerator to sterilize tools like the loop, which is shown here, a needle, which I'll show you in a minute, or forceps. Okay, and what you do is you stick the loop in there, get it red hot, and then bring it out, allow it to cool, and then pick up the bacteria on the loop, and then that loop is sterile before you use it to pick up the bacteria. You should know where the laboratory safety equipment is, and we do have a fire alarm. That's never been used in my lab. We have a laboratory shower too. If you ever spill acids on yourself, you can step under the shower. I've never had that being used. A spill control station, an eye wash station. You know, I don't think I've even had a student splash something in their eye and then use the eye wash station, but that's there. A fire extinguisher. And we have a fire blanket, which would be something, a container like that, where if somebody were to catch on fire, you could grab the blanket and throw it around them. All right, any question about the laboratory safety equipment? So generally, you can't do this for the first two labs, meaning lab 0 and lab 1, but pre-read the lab beforehand, and then you'll be ready to get go at the start of the lab. If you have any doubt, ask. And go ahead and read through the laboratory material. At the bottom we have the questions of the laboratory exercise. It's actually in a separate file. Let me show you in lab 1. Download worksheet lab 00 and write in your answers and then submit it in this folder. And I don't have the button to submit because I'm an instructor, but it should be over here on the right, or if you don't have items on the right, it'll be down on the bottom here. I've got a speed grader, which you won't have, meaning I can grade the assignments. That's it. You can download the document there. All right, so the final 2 pages go through safety rules, which I've already kind of talked about. And that's not at Clark, that's at Yavapai. I got to change that. And some safety procedures. I think I've already talked about that in the syllabus. So I won't repeat myself. All right, any questions? If not, go ahead and work on lab module 00. Fill in the worksheet. Maybe I should, well, never mind. If you have any questions, I will be here until, let's say, 3:45 to answer questions. And I will be here for as long as the last student is logged in. But if all students log out by 3:45, I will log out by 3:45. So if you have a question, make sure you log in before 3:45. Any questions? If not, go ahead and get started on the lab.